Hi, everyone. So I will introduce the next speaker. I don't know if I need it, but I can use it. Ronel Samuel from my group. Ron came to my group from to UB from the Chemical Institute of Technology in India, and he started his master's here as a master's student. His master's project was delivery hydrogels for delivery of proteins and growth factors for regeneration of salivary glands. Now, why we care about salivary glands is because many people cancer patients who receive radiation treatments and chemotherapy kill stem cells, and that's why people lose their hair. They also lose their salivary glands, and that has a lot of consequences to the point that they cannot eat. So they need to repair them. So after that, after finishing his master's thesis, doing that, he changed his topic into a stem cell-related topic, which is a big part of what my group has been doing for several years, and try to create progenitors, gland cells, and organoids. Organoids are small microtissues that have similar anatomy and function as the real tissue, and therefore can be used to both understand the biology of the system as well as for drug screening. And so this is what Ron will talk about. I will not say anything more. I'll let him tell you the story that he developed over the last several years. Ron?
Can you hear me? Perfect. All right, perfect. Thank you for that lovely introduction, boss man. So, as you probably are wondering, what are salivary glands? So, the entirety of my PhD has been on regenerating different strategies of regeneration of the salivary gland. And today I'll be sharing with you a developmental-based guide of how we regenerate the salivary gland. So what are the salivary glands? Well, your body has three major salivary glands: the parotid gland, the submandibular gland, and the sublingual gland. Their main function is to produce saliva. So if you're wondering what saliva is, it's that liquid in your mouth which you can feel right now. So what does saliva comprise of? Well, it's basically 99% water, but it contains 1% of proteins, which have very important functions. It contains proteins like amylases, which basically aid in the digestive process. It kicks it off. It also has proteins like mucins, which aid in the lubrication and swallowing of food. Saliva also contains proteins that aid in tissue protection and antimicrobial action, like histidins and cystatins. So why do we care about the salivary gland so much? Well, there are diseases, as the boss mentioned, that affect the salivary gland and affect saliva flow rate, and those are in two major categories. They are xerostomia and an autoimmune disease called Sjogren's syndrome. So xerostomia can be caused either via medication or due to any kind of head and neck proton therapy, which basically is for any kind of head and neck cancers. The autoimmune disease Sjogren's syndrome is basically when your immune system itself attacks and kills all the saliva-producing cells of the body. So all of these conditions together, they can lead to a loss in salivary flow and volume. So now that there's no saliva to basically regulate the oral microbiome, that can run wild, and you can have a whole host of dental and oral diseases. I'm just going to show you a few of them out here. So with an excessive buildup of dental plaque, you can get a disease called gingivitis, which, if not treated, can lead to advanced periodontal disease, which basically you lose your gums and your teeth. Loss in salivary volume is also associated with endodontal disease, which is called caries, which basically is a cavity in your teeth. So when you eat or when you drink, it causes severe discomfort. And lastly, I don't like this disease a lot. It's oral candidiasis. It's probably the worst one. It's a thin layer of fungus on your tongue. And that fungus basically anchors itself to your tongue and in between your epithelial cells of the tongue. and it's really hard to get rid of. If you try to get rid of it, it basically comes off as a sheet and also brings out some of your tissue along with it, so it leads to a lot of bleeding. So what is our strategy for salivary gland regeneration? Well, it's a development-inspired approach for the derivation of these salivary gland progenitors and also organoids from human-induced pluripotent stem cells. So you might be wondering, what is exactly human-induced pluripotent stem cells? Well, back in 2006, Dr. Yamanaka was able to take adult human tissue, mainly your easily accessible cells like your skin, your blood, and then using some reprogramming factors, although now you can do that even chemically, he was able to induce pluripotency to these cells and make them embryo-like. From these cells, once you have them, you can, using different biological and environmental cues, you can basically make any kind of cell type in your body. And this can be then transplanted back into the patient as a autologous source, so there is no chance of immune rejection that way. So in this schematic, our workflow will basically show how we generate these human salivary gland progenitor cells. And then, as the boss mentioned earlier, we're also going to show how we developed organoids from them. And organoids are important as they are 3D constructs that mimic tissue function and form. So it can be used for modeling diseases and also for any kind of drug discovery applications. What we'll end the talk with is, instead of showing us transplantation into the humans, we're going to show it into an immunocompromised mouse. So we're going to use these progenitors, transplant it orthotopically into these mice. So basically, we're going to transplant it into the submandibular gland of the mice. So with that being said, what is this developmental inspired approach? Well, we're going to see how the salivary gland develops and try to mimic it as much as we can. So the salivary gland emerges after embryogenesis. It splits into three main layers: the ectoderm, mesoderm, and endoderm, and the salivary gland is from the ectoderm. A closer look at the ectoderm will show you that this ectoderm folds in on itself to form the neural tube. And this gives rise to all your peripheral and autonomous neurons. And what we are left is the surface ectoderm. And that's the tissue from which the salivary gland also develops from. So this surface ectoderm, when localized to the head and neck area of the developing embryo, is known as the oral ectoderm. So let's see what this oral ectoderm does. To initiate the salivary gland, this oral ectoderm basically forms this prebud, which is a thickening of the epithelia, which invaginates itself into the surrounding mesenchyme, which then undergoes successive branching events to form this terminal, highly branched upside-down tree or a broccoli, which basically governs its function as well. So this is the embodiment of an adult gland, I'd say. So let's take a look at what are the different cell types in each of these compartments. So the gland basically has three major cell types, mainly the acini cells, out of which there are two types of acinar cells, the mucus cells and the serous cells. The mucus cells produce a more mucus-like saliva, and the serous cells produce a more enzymatic, watery saliva. They are flanked by these myoepithelial cells, which basically squeeze these serous cells to secrete the saliva into the lumen of the acinus. And the formation of these lumens is very important, as that's where all the saliva is collected. After which, the saliva is transported out by these ductal cells into your oral cavity. So this is the differentiation protocol that we came up with. It's a 46-day-long protocol, right from the induced pluripotent cells all the way up to mature acinar cells. So due to the lack of time, I'll just breeze through the first couple of sections. So initially, we wanted to get the surface ectoderm. So with the help of these molecules out here, we were able to block certain pathways that activated other types of cells and hence filter out them and get a mature surface ectoderm by day 10. After which, a 3-day induction with FGF2 and LDN basically gives the oral epithelium, that's the tissue layer from which the salivary gland develops from. After which, a 4-day induction with these factors basically gives us our salivary gland epithelial progenitors. So we tried to basically model the salivary gland epithelial progenitors after this initial bud, because this is the cell type which basically forms all the cell types contained in an adult salivary gland. So let's take a closer look at what this bud actually contains. So this bud is entirely SOX9 positive, and SOX9 is a transcription factor which has been shown to be the master transcription factor for all the salivary gland progenitor cells. And this paper really nicely shows it. So we tried modeling our cells after these SOX9 cells. So we took a look at our cultures, and we wanted to see if our SOX9 is also being expressed. And indeed, pretty much all our cells have SOX9. They also have E-cadherin and pan-cytokeratin, which are indicators of their epithelial phenotype. And a closer look at these cells basically showed that although all of them had SOX9, they did express SOX9 at a differential level. And here you can see with the E-cadherin and pan-cytokeratin staining, you can see the cuboidal nature, which is very reminiscent of epithelial cells. So now that we were sure that we had an epithelial cell population expressing SOX9, we also checked for different initiation markers, which are also of the salivary gland progenitor origin. So first up, we checked for the oral epithelial markers to see if they were downregulated. And indeed, SOX2 and PITX2, which mark the oral epithelium, were downregulated both at day 17, followed by an increase in SOX9, FOXY1, TP63, keratin 5, and keratin 14, which all together mark the initial salivary gland bud population. Now, as the gland develops, it splits into two parts, mainly the proximal progenitors and the distal progenitors. They're just spatially different. So the proximal progenitors, we checked for ASCL3 and keratin 8, which basically are progenitor ductal markers, and they were also upregulated at day 17, along with SOX10, which is a progenitor acinar marker. That was also upregulated at day 17. These two results together basically showed that our cells were primed to go down either of the two routes, but we will be focusing on these SOX10 progenitors because we want to get to the acinar cell type and not the ductal in this case. So how do we do that? We take these, now that we have these SOX9 cells, we want to transform them into SOX10 population of cells. So we do that by taking these two-dimensional cultures and basically embedding these cells in a dome made out of Cultrex. Cultrex is basically a hydrogel which is comprised of extracellular matrix molecules like laminin, collagen, intactin, and nitrogens. After we do that, we hit it with these group of factors for 8 days. And instead of these cells basically just clumping and forming one mass aggregate of cells, they were indeed able to reassemble themselves and self-assemble basically into these luminal progenitor organoids by day 25, after which a treatment of 7 days with these molecules out here was able to induce the SOX10 expression in these organoids. So by day 25, this confocal video basically shows the formation of these organoids, which are entirely luminal and also are basically show their hollow nature. And this hollow nature was also seen at day 32 in this confocal render, where we basically sliced off the top half of this part of the image to show the internal part, and we can see that it's entirely hollow on the insides. So now that we got these luminized organoids by day 25, by day 32 also we were able to retain this and also get SOX10 expression. And SOX10 was highly upregulated at day 32. And as you can see, the experiments are getting pretty long, whole month for one experiment. So we needed a system to basically track SOX10 in a live cell format. So what we did was we engineered our cells to express GFP whenever SOX10 is active. So GFP being the green fluorescence protein basically shows if our cells are green, we have SOX10. So we were able to actually modify our media to boost this SOX10 GFP activity, and hence we arrived at this composition of media for the maximal amount of SOX10 being produced. So now that we have our SOX10 progenitor organoids, our goal was to mature them fully into saliva-producing salispheres. So what is a salisphere? A salisphere is basically an organoid which has MIST1 in its nuclei. MIST1 is a nuclear-located transcription factor that defines all acinar lineages of cells. It should also have NKCC1, which are these outer membrane located, which is known as basolateral ion channel proteins. So they basically mediate the co-transport of sodium, potassium, and chloride ions. It should also have aquaporin 5 on the apical membrane, which is the inner membrane. So aquaporin 5 helps mediate the transport of water, and saliva is 99% water. So having these molecules out here on the internal membrane is very key to its function. These also should also have eq-adherent adherence junctions. They are basically junctions which bind neighboring cells' plasma membranes together, hence forming and maintaining the structure of this organoid. They should also have zonular colloidins 1 tight junctions. These act as seals between the cells and hence show as a sign of polarity in our cells, that the saliva is being transported only in one direction towards the acini lumens. And finally, we also wanted our salispheres to have amylase 1, which, as I said before, is a key enzyme present in saliva, and it basically starts off the digestion process. So we checked for all of these markers, and first up, we checked for MIST1, and we noticed that our organoids have MIST1, and they express MIST1 to different degrees within the organoids, but they're all having MIST1. They also have NKCC1, and they have NKCC1 towards the basolateral membrane, which is very key to its function. We also were able to see aquaporin 5, and especially we were able to see aquaporin 5 out here on the internal apical membrane, showing that it's correctly localized to where we wanted to see them. We also had eq-adherent adherence junctions and also ZO1 tight junctions. And finally, we also were able to see in this video the presence of amylase. So this video nicely shows that the 3D render of our organoid, and we were able to see amylase in our organoids as well. So, however, what we did notice is that this amylase was sequestered towards mostly the basolateral side and not present in vesicular formats near the apical side, showing that it's probably not fully mature. So we would say that our organoids at this point were able to mature to a certain point, but not fully. What we also saw was the presence of these internal F-actin rings, which together with the ZO1 activity, we were able to conclude that our organoids were in fact polarized. So now that we were able to create these acinar specific salispheres from our epithelial progenitors, our initial hypothesis also said that maybe these cells can also give rise to duct cells, creating a sort of bipotent progenitor phenotype. So to address that, we implanted our cells into a mouse immunocompromised mouse model, along with the same hydrogel, Cultrex, and prior to the transplantation, we labeled our cells with M-cherry, so basically our cells fluoresce red. So that way we can easily distinguish our cells from the mouse cells, because the mouse cells will not fluoresce red. So initially, in the mouse tissue, you can see very nice arrangement of these acini. So here, MIST1 being an acini marker, you can see that it's nuclear located very nicely within the mouse tissue. And the cells which have this arrangement but don't have MIST1 are basically ductal cells. So in our system, we also see our transplanted cells after 6 weeks were able to form these acini structures, which are very reminiscent to this, and also ductal structures reminiscent to this. At 12 weeks post-transplantation, we were able to see more of these structures being formed with more amount of acini, showing that these cells were able to survive, engraft, and differentiate into these adult acini, basically secretory markers, basically. And to address our initial hypothesis, we basically stained our cells with both MIST1 and keratin 818, which is a ductal marker. And we were able to see expression in certain cells with both keratin 818 and MIST1, showing that there is a bipotent progenitor phenotype that basically gives rise to both ductal and acinar cells. So now that we were able to prove our initial hypothesis, right, what we also hypothesized was that whether these cells, once transplanted, can be functional. Can they become functional? So in this section of this mouse tissue, you can see that we checked for amylase, and you can see amylase is present in these vesicular formats towards the internal apical side of these acinar cells. And these yellow regions are basically your acini, whereas the regions which don't have amylase are ducts. So we checked for amylase in our transplanted tissue as well. And 6 weeks post-transplantation, there was very less amount of amylase present in the cells, and most of it was also cytoplasmic. However, 12 weeks post-transplantation, we start to see a more amount of green, which is amylase, but again, it was mostly cytoplasmic in nature, which basically tells us that our cells were able to, again, survive, engraft, differentiate, and also produce functional proteins, but not in its correct format. So it should be in these vesicular formats, but here it's mostly a cytoplasmic stain, telling that most of the cells which we transplanted are not fully functional by 3 months and probably need a little more time to differentiate fully. So in conclusion, we were able to generate a SOX9 positive salivary gland progenitor phenotype, from which we were able to derive a SOX10 progenitor phenotype, which are pro-acinar organoids. And then we were also successfully able to mature them to form these MIST1 and amylase1 positive salispheres. We also were able to transplant these progenitors into the immunocompromised mice to basically show that they were able to survive, engraft, and differentiate into the different adult salivary gland lineages, hence showing its bipotent phenotype prior to implantation. Our future work basically contains this same transplantation, but with the day 46 salispheres, as they already have the proper shape prior to implantation, so we might see a faster regeneration. And also the use of NRG1, which is a neurotrophic factor, to decrease SOX10 activity and hence fully mature the salisphere to respond to cholinergic signals to show functionality of these organoids in vitro itself, so it can be used as a drug testing model. So with that, I'd like to thank again my advisor, Dr. Andreadis, for funding me for so long and my work as well, and also giving me this opportunity to talk to you about my research. And also my lab mates, especially Dr. Pedro Ley, who works with me, and also Laura Sherwood, who will be taking this project on. And also my collaborators, Dr. Olga Baker and Dr. Ki-Hun Nam, and especially Dr. Ki-Hun Nam, who actually did all the mouse surgeries for us. And finally, also our funding sources, the NIH and NIDCR. Thank you for listening.
Thank you for the presentation. So other than the ductal cells, aren't you missing the cell that squeeze the saliva out? So do you have any plans of that?
So I anticipated that question coming somehow. We do get a tripotent progenitor phenotype. So here you can see these, wait, it's not out here. How do I get it back on?
Did you accidentally tap this screen?
No, I don't think so. Oh, no. Maybe tap. Oh, there, it's inside. So we do see this in our transplanted cells. Here you can see these little white cells out here which surround this mass of cells. They are stained for alpha SMA, which is a myoepithelial cell marker, and they also stain for M. cherry. So this is basically our own cells, which are also able to differentiate into these myoepithelial cells. I just didn't include it because of time.
Okay, no problem. Thank you.
Yes.
Great talk. I guess the in vivo transplantation is a big challenge. You presented obviously positive results today. What were the big negatives? I guess the choice of progenitor, vascularization, and I noticed of course, you presented your salivary organoids are kind of inverted maybe, or I guess you'd have a branching duct coming from it if you're squeezing saliva into the duct, but immediately I thought they were inverted and you'd want the saliva to go outwards.
So, if I can show this. So our organoids which we were able to make, they did show this activity where, like, amylase is towards the outside membrane, where it should be towards the inside membrane. But yeah, I guess we have to make them fully mature as to, as I said, with the NRG1. If we can properly functionalize the tissue and then transplant it, we probably get a better, result with that.
But then they probably won't grow as much.
Probably true, because they lose their progenitors. Yes.
All right, was that crowd again?